Welcome to Cancer HealthCast, where science is driving hope. I'm your host, Nikki Henderson. Today, I'm joined by Dr. Clint Allen, who is a senior investigator in the Center for Cancer Research at the National Cancer Institute, and Dr. Scott Norberg, who is an associate research physician in the Center for Cancer Research at the National Cancer Institute. Today, we will be discussing a recent study about a disorder caused by the Human Papilloma Virus, how it impacts a patient's life, and the role technology plays in treating the disease. The trial was conducted as part of a cooperative research and development agreement between NIH's National Cancer Institute and the biopharma company Precigen. Hello, Clint and Scott. Thanks so much for joining us on the show.
Thanks for having us.
Yes, thank you.
To start off, Clint and Scott, can you both talk a little bit about your roles at NCI and how they support the agency's overall mission?
Dr. Norberg.
So I'm an associate research physician and a clinical investigator in the NCI Center for Immuno-Oncology. And my role is to conduct a novel, usually first in human phase 1 clinical trials, testing treatments for premalignant and malignant conditions. So I have a special interest in virally associated diseases and cancers, including my clinical and translational research interest in recurrent respiratory papillomatosis. So my goal is aligned with the institutes and our branch in that we really try to understand either rare diseases, understudied patient populations, and also try to develop novel treatment options and diagnostics for these patients.
This is Clint Allen. So I'm a senior investigator in the National Cancer Institute, and I head a large clinical and basic science research program that focuses on basically ENT neoplastic disorder, so ear, nose, and throat. I'm a board certified otolaryngologist, and my research program works on head and neck cancer, both HPV positive and HPV negative, and then more recently on this disorder we're going to talk about today, recurrent respiratory papillomatosis. And I think Scott and I have many of the same goals. Our goal is to try to make patient care better for disorders that we think have room for improvement, and particularly to study rare disorders that are very difficult to study anywhere else because of difficulty with getting funding or because of difficulty with amassing enough patients together at a single institution to be able to do meaningful clinical trials.
Well, Clint, before we get into your recent study that was published on recurrent respiratory papillomatosis, or RRP, can you describe the pathophysiology of this disorder, what it leads to clinically, and how it can affect a patient's life?
Yeah, so recurrent respiratory papillomatosis is directly caused by chronic infection with HPV type 6 or 11. These are different HPV types than cause cancer, but these are the same HPV types that can cause papillomatous lesions elsewhere, like on the skin. And for reasons that we don't completely understand, a subset of people with completely normal immune systems otherwise, upon exposure to these HPVs, will develop a chronic infection. The infection is in the mucosa or the lining of the throat, voice box, and windpipe. And the chronic infection can cause these papillomatous growths. And because of where the growths occur, if they're on the vocal cords, they cause profound voice disturbance that we call dysphonia, really to the point that it inhibits people sometimes from working outside the home, has severe detrimental effects on interpersonal relationships with friends and family. You know, people can't even order food at a drive-through because it's so difficult to understand them, difficulty talking on the phone, etc. When the papillomas become big enough, really anywhere in the voice box or windpipe, they can actually block the airway and cause difficulty breathing.
Now, Scott, can you please tell us more about your RRP study and outline some of the key objectives?
One thing to add to what Dr. Allen just mentioned is that the current standard of care is clinical interventions to remove the papillomatous disease burden that comes into the voice box and the trachea that affects voice and breathing. And so, as you can imagine, the surgery itself doesn't address the underlying cause, which is chronic HPV infection. So really, there's been a push in the field to try to move away from surgical treatments of this disease to medical treatments. So the study that our group conducted was of an agent called PRGN 2012 that was administered in the adjuvant setting. And really, the overall goal was to try to eliminate the need for further clinical interventions following this one-time treatment course. And so, kind of in summaries, these patients, the patient population had severe RRP, which we defined as the need for 3 or more interventions in the 1 year prior to enrollment into the study. And the way that this treatment was deployed is that patients underwent an initial standard of care surgical debulking treatment, followed by their first administration of PRGN 2012. They then received 3 subsequent administrations of treatment on days 15, 43, and 85. And at that 43 and 85 time point, patients that had disease that looked to be amenable to further debulking underwent another procedure with the goal of trying to maintain a state of what we call minimal residual disease. And really the goal for that is that we felt like this therapy had the best chance of working with the minimal amount of disease during this one-time treatment course. There were patients that didn't have disease that required that kind of intervention on days 43 and 85. So they simply just received the 3rd and 4th dose of drug at that time. And the overall objective of this study was to determine whether or not, following this one-time treatment course, patients could go without requiring any further clinical interventions to control disease. And so this was our definition of complete response. And we also looked to see whether or not there were patients that had any kind of decrease in the number of interventions following treatment of the one-time treatment course, which again is really an important goal in this patient population. In addition, safety, of course, was an important aspect of the study. So in general, we found that the treatment was well tolerated. There was no dose-limiting toxicities, no serious adverse events, or grade 3 or higher treatment-related adverse events. And really the most common side effects were mild and self-limiting. They included injection site reaction, fatigue, chills, fever, and muscle aches. No patients had to stop the drug due to untoward side effects, and all patients were able to receive all 4 doses of the drug. And what we found remarkably is that a majority of the patients, 51% of them, didn't require a single intervention in the 1 year following treatment. In addition, a majority of patients, 86%, had any kind of decrease in the requirement for clinical intervention in the 1 year following treatment. And importantly, a majority of these complete responses that we saw were durable, and a majority of them were still ongoing at the time of the study publication. This data was used as a basis for the biologics license application of this agent to the US FDA. So really, we hope that this is a prelude of being able to use medical treatments in what was once really considered just a disease that was treated with surgery.
That is wonderful news. It really is, and quite remarkable. And it seems like there's going to be a lot of, like you said, promise with this new treatment with the medicine rather than having to do surgery. Well, Scott, I'm going to stay with you for my next question. I wanted to know, what role did technology play in developing this new gene therapy to treat RRP? And can you discuss some of the pros and cons of these types of interventions?
Yeah, so the drug uses a gorilla adenovirus-based technology where it's a adenovirus that actually specifically infects gorillas. And so because of that, humans don't have a lot of pre-existing immunity towards this virus. And the way that technology was used is that the virus was modified in order to remove certain components of the virus that make it active and cause an active infection and cause it to proliferate. So those were removed to make the the virus safer for human administration. And instead, what was placed into it was actually DNA sequences that encode for proteins that make up HPV-6 and 11. And so these are the proteins that then can train the immune system to be able to recognize and eliminate HPV-6 and 11 infected cells. So really the advantage of the platform, as I kind of mentioned before, is because this is a adenovirus that really only infects gorillas for the most part, is that there's very little existing immunity in human population, which means that the immune system would not eliminate or neutralize this treatment, soon after administration. And this also allows for the drug to be administered multiple times without the immune system neutralizing it, really in a sense, neutralizing the treatment. In addition, the platform also allows for large quantities of DNA sequences to be put into it. And this allows for a patient's individual immune system to really be trained on the HPV-6 and 11 proteins that it needs to target. And so, this provides a lot of advantages of clinical activity. But the one thing that we found in the clinical trial was that this treatment was also well tolerated and quite safe.
That's wonderful to hear as well. Thank you so much, Scott. Clint, can you please explain the rationale for developing immunotherapies directed at inducing an HPV-specific immune response?
Sure. So about 5 years ago, we began considering different forms of immunotherapy to use in clinical trials for patients with recurrent respiratory papillomatosis. And the initial clinical trials that we did focused on the use of immunotherapy in the category of immune checkpoint blockade, which is now, widely used in numerous different kinds of cancer.
Mhm.
And the rationale here was that immune checkpoint blockade would unleash the activity of immune cells in the body, and that those immune cells would be smart enough to go specifically after HPV and the papilloma. And what we observed in those clinical trials that are all published is that we certainly saw shrinkage of disease to some degree with the treatment, but the treatment was not making everyone's disease go away. We weren't seeing what we would consider to be deep or complete responses. And immune checkpoint blockade as a non-specific immunotherapy carries risk of activating the immune system against something other than the papilloma, like an important organ. And that risk, combined with the lack of profound, deep, or complete clinical responses, really made us change gears and want to pursue something that was much more specific. And that's where this idea was born, that if we could find ways of specifically activating or expanding the immune response against HPV, that may have a much more positive impact in a disease that, as far as we know, is purely caused by the chronic infection with HPV.
And also, Clint, I know that NCI's last mile initiative utilized self-sampling technologies to screen for HPV. Can you tell me how were the patients with RRP diagnosed?
Yeah, it's an interesting question, because diagnosis of RRP is a clinical diagnosis. And
Mm.
right now, diagnostic criteria actually don't include testing positive for HPV-6 or 11. If a patient has one or more papillomas found to be, in the throat, voice box, or windpipe, the papilloma is removed. The diagnosis of papilloma and not something else like cancer is verified by a pathologist. And then if that papilloma comes back, by definition, that's the clinical diagnosis of recurrent respiratory papillomatosis. I will say that there is now a commercially available test that is available through Cincinnati Children's Hospital that allows CLIA certified verification of HPV-6 or 11 within a papillomatous lesion. There were many commercial tests that were available before that, but those tests did not distinguish between HPV 6 or 11. They would just tell you if the lesion was positive for 6 or 11. But again, the confirmation of HPV 6 or 11 positivity is not currently considered one of the diagnostic criteria for recurrent respiratory papillomatosis, but that may change in the future.
And Clint, can you please describe how you see the care of RRP changing over the next few years? Is there a possibility that patients will be treated more so with medications rather than having to undergo surgery?
Yeah, I think that's certainly our goal as a field of investigators studying RRP. As Dr. Norberg described, these procedures that up to and now still remain standard of care treatment for RRP. While the treatments are necessary to reduce the size or debulk the papillomas to try to help maintain an open airway and some usable voice for patients, the procedures themselves can actually lead to scarring and real long-term dysfunction of the voice box. And so it was really a necessary evil. It's been a necessary evil to do these procedures. And it's not unusual for an individual patient to have required hundreds of procedures over their lifetime to keep their papilloma in check. And so for many years, we've been searching for ways to try to address, the underlying cause of RRP, which is this chronic HPV infection, with non-invasive means. And I think clinical trial results like ours are a significant step towards that goal.
Right, I agree. And wow, you said hundreds of surgeries for some of these patients. That is very difficult for someone to undergo. So that would be wonderful if the medications would be able to treat this. That's just wow. That's a lot to digest. Well, Scott, can you also talk a little bit about the need to develop treatments for pulmonary RRP?
Yeah, absolutely. Despite our, very positive clinical trial results, there still remains a major unmet clinical need in addressing pulmonary RRP. And pulmonary RRP occurs in about 10% of the patient population of RRP. And these patients also have a significantly increased lifetime risk of that disease becoming malignant. It can lead to recurrent obstructive pneumonias, infections in the lung, respiratory failure, and even death, unfortunately, in some patients. So right now, the only really effective treatment has been surgical resection. But again, it doesn't address the underlying cause of the chronic infection. And it's also not feasible in patients that might have multiple lesions throughout multiple lungs, lobes of their lungs in both lungs. And so, medical treatments to date that have been tested, including the PD-1, PD-L1 checkpoint blockade, bevacizumab, and the DNA immunotherapies, they really haven't shown a consistent ability to control pulmonary disease. There's also case reports testing different types of chemotherapy agents and even radiation treatment. But again, these results have really been pretty dismal. So we're actually studying the use preclinically and then hopefully in the near future clinically of adoptive T-cell therapies to be able to be used as a treatment for this patient population. And a lot of it stems from what we have learned thus far of using those same types of therapies in HPV-associated cancers, such as cervical cancer. And so this is an active area of research for our group. And again, with the same premise and goal in mind of trying to replace a surgical treatment with the medical therapy.
Well, this is a question for both of you. Are there any future initiatives that either of you is excited to talk about?
I would say, I think, going along the same kind of premise of, really changing the overall mindset of the field where we are looking to really, in a sense, put medical treatments as the front line rather than necessarily surgical treatments. And we just think that there's so much more long-term potential benefit of kind of changing the mindset of treatment for RRP, really towards a medical treatment, because it is a medical disease that, to date has been cared for by surgical interventions. So I think that the whole kind of mind shift and, moving away from what has been done traditionally for really decades and even beyond that is really kind of an exciting, prospect.
Yeah, I would agree with that. And I guess the way that I would phrase it is, the care of patients with head and neck cancer has required multidisciplinary team care for decades because of the expertise of the surgeons and the expertise of the medical oncologists who administer the systemic agents. And it's going to take a real paradigm shift in the way that patients with RRP are cared for. And that shift is going to be, the development of multidisciplinary teams consisting of otolaryngologists as the surgeons and medical oncologists that have experience and expertise in administering systemic, agents, and in this case, potentially these DNA immunotherapies. I agree with Dr. Norberg. That's a challenging but very exciting path forward that I think will really result in tremendous clinical benefit for these patients that to date have really had no other options.
Wow, that's wonderful. That's wonderful. I hope that that becomes the case, that medical treatment will be the primary treatment. Well, before we conclude, I just wanted to check with either one of you to see if you have any final remarks or comments that you would like to leave with our listeners.
I would say that I think the National Institutes of Health is probably one of the few, if not the only institution in this country that could have performed this type of research. This is a relatively rare disease, and it's difficult to get enough RRP patients together at one institution to be able to do these types of clinical trials. It's very typical for an adult otolaryngologist in even a large academic institution to care for 5 or maybe at most 10 of these patients in their practice. So having the availability of the NIH Clinical Center as a site and being able to support patient travel to be able to bring patients in from all across the country for these clinical trials, I think is a very unique and powerful feature of the NIH research program.
Okay, great, great. Anything else?
Yeah, no, nothing for me. I think Dr. Allen said it great.
Well, this was a great conversation. It was very informative. So I thank both of you so much for sharing the findings from the RRP study and just discussing how tech plays a role in treating this disease and talking about the medical treatment, how that is the desired end goal. So I appreciate you both taking time to speak with me, and I hope that you both have a wonderful afternoon.
Thank you.
Thank you.
Thanks for listening to Cancer HealthCast, a production of GovCIO Media and Research. To explore our content, visit our website, govciomedia.com. Keep an eye out for new Cancer HealthCast episodes every month. And if you like what you heard, leave us a review on the podcast platform of your choice. Have a topic you want us to discuss? Contact us at newsletter@govcio.com.